Welcome to Bio class. Today we will discuss DNA fingerprinting. DNA fingerprinting, here I think everybody can understand that is no relation with the fingerprint, but here the DNA fingerprinting, this word we are using, that unique combination of the DNA of any particular individual, and from that we can detect the particular person from the millions. DNA fingerprinting is a technique. It means a technique to identify a person on the basis of their DNA specificity. It means the DNA, it can be, similar sequence and 99.9%, but still there are difference. But it is a very complicated, because the DNA, our DNA is very long. Here is a special technique it is applied, so that very easily we can detect that the unique features which is found in the DNA, and we can identify the person. First detected by Sir Alex Jeffrey, got the Nobel Prize. Here is Sir Alex Jeffrey, and the first criminal case and his DNA is in Scotland. His DNA is detected in. Here is the victim. So the first case it is, and here is Sir Alex Jeffrey. Now, what is the principle of DNA fingerprinting? Already it is told that it is a very long molecule, and huge combinations. Combinations unique, but it is very difficult to get that the sequence very easily. It is so long. But here the process which is applied, that we have to learn. We know that in DNA, the huge portion, it is introne in between gene. Suppose this is the exon, and exon portion is only 2%, and 98% is introne. That already you know. Now, in our introne, there is some special character, and we are using this character here. In introne, there are certain sequence. Suppose one certain sequence is there. This sequence is repeating many times. Okay. The same sequence is repeating. Okay. So here it is a sequence. I'm not writing the sequence. It is there, and it is repeating. Suppose here it is repeated 5 times. Okay. This 5 is the unique number of that person. It means that the person to person, this number, the 5, or maybe 45, maybe 31, maybe 66, whatever the number of the repeat, that will be unique. That will be the different, and in that we can easily understand the person. So variable number, tandem repeat. Variable number what? This is the variable number. Just now I told that 31, 42, 56, 120, whatever. This variable number, tandem means one after another, and this repeat is there. Where repeat is there? In introne. Where repeat is there? In between 2 gene. So that is the concept of the DNA fingerprinting that we have applied here, and this feature, it is there in our genome. It is there in between gene, the introne part, which is normally, you know, that it does not produce any protein. It is junk. This area also, here it is used in this detection of the DNA fingerprinting. Now in VNTR, the DNA tandem repeats, there are few terms just you see that the repeats, the size of the repeats, if it is 1 to 9 base pair, it means that less number here. Here it is 5 in between. Okay, so this is called micro satellite. It is also termed as ASTRs, short tandem repeats. So how many repeats are there? 1, 2, 3, 4, 5, 6. Here the 6 repeats. So this number is unique. Already I discussed that mini satellites, which is VNTR, variable number, and here it ranges 10 to 100 base pair. Suppose here 11 it is nucleotides, and it is 4 times the same sequence. It's repeating one after another. Okay, there is 4 repeats. So another person, it will be some other number, 8. Another person can be 86. In that way. Another is that macro satellites, which is size is more. So on the basis of the size, the micro, mini, and the macro satellites here it is, and there. Now come here in process. How we can do this process? Here is the sample. It is written blood sample, but this sample can be blood stain, can be semen, can be hair root, saliva, tear, any skin part, even that the person's victims, the clothings, even it is several years old, then also this process will be successful. Okay, so the first DNA, it is will be extracted from the blood, from the sample that already I told, and after that the DNA it is fragmented. Here we will apply the restriction enzymes. In biotechnology, that everything it will be discussed. Restriction enzymes. This restriction enzyme, when it will be fragmented, means the gene and the non-gene, the portion will be fragmented. Then after that we will use the gel electrophoresis. The detail I'll show you, that gel electrophoresis, how it works, and we'll get that this type of the band like structure. So we will make it permanent with the help of southern blotting system, and after that this can be detected by auto radiography and some other means. Nowadays it is there. Okay, here is a gel electrophoresis, agarose gel. Look at it carefully, listen this part. This is the porous gel. This gel we are making from the algae agar-agar. We are removing the agaropectin and making it agarose gel to the process. And here you can see that this is called well. In this well we are here dropping this sample, that after treating the restriction enzyme, that sample from the blood and the victims, suspects, those who are there. So one after another here we are dropping. Here it is negative, it is electrode, it is there, and here it is positive. DNA, you know that negatively charged, so it try to move. See that the movement, DNA fragments move through the gel towards the positive electron. But what is the problem? That the longest fragments, they cannot move so fast, so they are making the band in the negative sides earlier. And those who are shortest one, they can move there, so that these bands are, it makes it different. See that variable number, it is more, then the fragments will be heavier, and where the variable number is less, so that it is shorter and it can come quickly. This things I want to show you in a video. See that. Look at here, different sample it is placed in in electrophoresis. So after that it is covered and the power is given. Then a negative electrode, now it is moving towards the positive sides, and it forms that kind of the band structure. See that those are having the mode repeats there at the back, and see that how it can possible here. This gel is porous. You just imagine that this is the DNA fragment. If it is longer, if it is very long, so that the movement will be not fast, whereas in sorted they can move very fast with in this porous gel. That is here it is shown very nicely. Just you can see that here ultimately we get the band like structure. When we got that band from the gel, after that, the gel it is not permanent. We have to make it permanent with the cellulose nitrate paper, and here is the gel. So this gel set, and here is the stack of papers, glass and some other, but main thing here the gel and the cellulose, nitrocellulose paper, photographic plate which can uh, take the image. And here the paper stacks, and here something heavy which is more than 500 gram weight. So in this way it can be kept for long time, overnight it can be kept, and it is baked in 80-degree for the 2-3 hours to fix the DNA permanently on the membrane. Okay. So here is the steps. First, isolation of the DNA, digestion by the restriction endonuclease, separation by the fragmentation gel electrophoresis, then transferring the blotting that in membrane, nitrocellulose membrane. Okay, up to here we have completed. Now, hybridization using labeled VNTR probe. Actually, the probe, that concept will get afterwards. The probe, it means that some complementary stand which can detect. It can be radioactive, can be fluorescent. Nowadays it is used. That I'll discuss in biotechnology, that part. Detection of the fragment in autoradiography. After autoradiography indicates that gets the similarity or dissimilarity, and how the autoradiography that it can do. Let's see its result, how we can at last we can detect. In your book it is given very nicely. Here it is a result. Here we have got this is from crime scene. We have got, suppose blood or something. So here we have got that there are the two chromosomes, I mean homologous chromosome. So it indicates that from the parents, one from father and one from mother. It will not similar because two different body. The number of the short tandem repeats here it is. Look at here it is 11. So 11 when it is, so it is heavy, so it makes band here. All right, and here chromosome 7. Chromosome 2. Next one you can see that that repeats, the number of repeats. So here number of repeats 11, number of repeats 9, here number of repeats 8, number of repeats 7. So in that way it is arranged. 8 is here, 7 is here. Here number of repeats 5, and the number of repeats 2. So 5 here, 2 here. So this is from the crime scene, and these are the suspects. So suspects there what kind of band that is there in the chromosome 7, 2 and 16. So look at here the individual A. I don't think it is a matching. but individual B is matching because here it is. You can see the same type of band which can. So here the advantage is this analog type of structure. From that you can easily able to understand, easily it can be detected that the person which is victim, and easily you can detect that this is the crime scene. Whatever DNA we have got, it is matching with this person. So that is the unique of this DNA fingerprint. Nowadays it is more advanced, and easily we can do that. In biotechnology, actually this part it should be given to the in biotechnology, that so many things are, suppose restriction enzymes and other things are there in the biotechnology. So we will discuss after that. Okay, thank you.